Great. Thank you, everybody, for coming. It is my great pleasure to introduce Dr. Nicole Mideo from the University of Toronto. Nicole did her, let's do the career bit, shall we? So Nicole got her PhD with Dr. Troy Day at Queen's University before moving to the University of Edinburgh, where she worked as a research fellow with Sarah Rees. And then she came to work at Penn State with Andrew Reid. And that was very lucky for me because I was just starting out as a beginning grad student at the time. And in addition to bringing her technical nous and synthesizing mind to the floor, she also taught me how to ask and answer a good question, and was a good model of one way of being a scientist. It was possible, she taught me, to ask questions with reckless abandon and to portray your passion for your subject. And I'm sure we'll see that in evidence today. So I will turn over the floor to Nicole Mideo.
Thanks, Nina. Can you all hear me in the back? Yes, thumbs up. This is the weirdest geometry for a seminar room I've ever been in, so I have no idea where to stand or what to do with myself, so apologies in advance if I am pacing too much. If I am, I don't know, yell at me or something. I will try to not disappoint you with the reckless abandon and enthusiasm that Nina was speaking of. I wanted to start my talk today. First of all, thanks for having me here. I've had a really great day full of super stimulating conversations, and I look forward to more conversations tonight and tomorrow. And I wanted to start by giving you a kind of a really brief overview to the kinds of questions me and my lab like to answer. And so we kind of asked, I'm going to start with reminding me to tell you that most of the work I'm going to talk about is work done by my amazing postdoc, Dr. Megan Gresher. So most of the cool stuff I'm telling you about is her work, and we've worked in collaboration with a number of people around the world on the projects that I'm going to tell you about. Now I will tell you about the big, broad question that my lab is interested in, and that is, why do some parasites lead to severe infection and others don't? And so, I'm kind of a evolutionary ecologist, and as evolutionary ecologists, we're interested in variation. So this is the kind of variation that I'm interested in, the variation in the severity or infectiousness of disease that different parasites cause. And I can take us on a tour through the suite of organisms that lead to infectious disease, and we can look at the ones up on these slides and think about the massive variation we see across different kinds of organisms that cause disease. Mostly I think about variation within a single parasite genus. I think about Plasmodium, which is the etiological agent of malaria, and I often think about a single species of malaria parasite and understanding variation across genotypes of that single species. And so we know a lot from biomedical research about the molecular pathways that lead to disease, but using our kinds of tools, ecological and evolutionary tools, we can ask questions about the particular organisms and their traits that lead to the disease symptoms. So I kind of think about three different kinds of questions here. I think about those ecological ones, the proximate explanations for variation in disease. So again, what is it specifically about the traits of parasites that lead to disease symptoms, and how do those traits vary across different parasite genotypes? I think about the sort of evolutionary questions here. Why are those traits the way that they are? Why aren't they some other way? What are the selective pressures acting on these parasites? And then I think about the consequences of all these things, either epidemiological or evolutionary consequences. Variation is the raw material of evolution, so can we predict how parasites will change in response to different sorts of pressures? So those are the really broad, vague sorts of things that we're interested in. Today I'm mostly going to talk about this box, but you'll see that obviously ecological interactions and consequences also come up in what I'm going to talk about. So, really brief outline. I'm going to tell you about a key trait of malaria parasites that I think is really interesting and important, and that's transmission investment. And then I'm going to walk you through kind of the logic of my work and the work of Megan, my postdoc, over the last several years to quantitatively titrate the influence of ecological processes at all of these different scales on the evolution of this trait of malaria parasites. Okay, so I'm quite interested generally in life history theory and the sort of resource allocation trade-offs that organisms face. So organisms have a limited amount of resources to allocate into different important life functions, so growth and reproduction. And roughly speaking, resources that you shuttle towards one of those important life functions you don't have available to shuttle to the other one. So organisms face these trade-offs in allocating the resources between these different parts of their life. I'm going to make the case that malaria parasites face the same trade-off. So these are trade-offs that are common to all sexually reproducing organisms. So a malaria parasite in the blood phase of an infection in a vertebrate host, these blue things here are my malaria parasites. They invade these host red blood cells, replicate inside those blood cells, and eventually burst out of those blood cells. And then those progeny parasites can go and invade red blood cells again, begin the cycle anew. Sometimes, instead of following this developmental pathway, a single infected cell can produce a single one of these, either male or female gametocytes, which are the transmissible stage of the parasite. Those are the only stages that, when they get into the mosquito, they can generate an infection within a mosquito vector. So these guys, if these guys get ingested by a mosquito, they just get metabolized, they don't do anything. You need these ones to be able to infect a vector and ultimately infect a new host. So this seems pretty analogous to that growth versus reproduction trade-off I presented for squirrels and trees. You can grow an infection, you can maintain an infection, or you can generate the possibility of reproducing an infection, generating a new infection in a new host. So what's very strange for malaria parasites is they seem to allocate very little towards reproduction. They allocate very little towards transmission. We sometimes call this conversion rate, so if I slip into calling it conversion rate, that's why. I'm trying to call it transmission investment. And if you look at sort of the relative abundance of these red blood cell infecting stages versus these transmissible stages, it's about 1%. Don't get too caught up on this number. My postdoc Megan has also shown quite nicely that the way people have gone about measuring transmission investment is biased by a bunch of processes, so we don't have a good grasp on what transmission investment is in natural infections, but we know that it's low, it's quite low. Okay, why do we care about transmission investment? I'm telling you that this is an interesting trait. Can I convince you of that? Hopefully I can. Obviously, how much they're deciding, how much parasites are allocating towards transmission versus that asexual replication cycle is going to determine probably the severity of infection. Those asexual parasites are the ones that are infecting red blood cells, bursting blood cells. That's not good for a host. So the more that's invested in this asexual proliferation means, all else equal, the more blood cells you're going to burst in your host. So transmission investment is going to influence the severity of infection. It might also influence, it's going to also influence infectiousness, but it might also influence things like the efficacy of drug treatment. So anyone who works on malaria in this room knows Nick White is sort of the godfather of drug resistance in malaria parasites. And one of the things that determines treatment efficacy is how many parasites are there when a patient comes in to be treated. These drugs work in this kind of nice way to model it. They follow these log linear declines. Parasite densities follow these log linear declines when you start treatment. So if you start at a higher density, it's going to take longer to clear all those parasites. Okay, so I think this trait is important. It's intellectually interesting, but it's also important because it's going to influence the severity of disease, infectiousness, although both of those things in a way that's a bit complicated, as we'll see, and it might influence the efficacy of drug treatment. Okay, so as a PhD student, I was really interested in this idea that malaria parasites actually invest very little in transmission, and that seemed weird from a sort of evolutionary perspective. Why are they putting so much energy and resources into these host-damaging asexual parasites and so little into the opportunity to generate new infections? And in sort of evolutionary epidemiology, generating new infections is the currency of fitness for parasites. So I explored this idea analytically as a PhD student, and so what I mean by that is you can come up with this expression, R naught. So this is in evolutionary epidemiology, it's the number of new infections generated by a single infected individual in a totally susceptible population. And this is our metric of fitness for parasites, generally, frequently. So how many new infections can one infection generate? And so this R naught expression, I'm not going to belabor the math here, but the only thing that's important to know here is it basically tracks all of the processes that go from one infected host through the vector to another infected host. And so you can ask, using calculus, well, how does transmission investment influence these different processes, these different parameters, and what does that do to R naught? Increasing transmission investment, does that increase or not through some of these parameters or decrease it? And there was a few hypotheses in the literature about why parasites might invest very little in transmission. Here are some of them. So the first one, mosquito mortality, maybe if parasites are investing more in transmission, then they're making more transmission stages are getting into the vector, maybe that's bad for the vector, maybe the vector can't survive. Similarly, if parasites are investing more in transmission, maybe that's generating a stronger immune response in the host, and that'll slow transmission to the vector. So these were two hypotheses in the literature to explain this observation of low numbers of gametocytes and low investment in transmission. It turns out that neither of those hypotheses can explain low investment in transmission. They can explain producing low numbers of gametocytes, but that's a different thing. And I'm going to try to make clear why that's a different thing. So basically, the approach here is treating a host like a bag of blood. That is how I simplify you all in my head. You are a bag of blood. And what should the parasite do? So in this case, the parasite has infected every blood cell in the circle. There are 8 of them, and has converted 2 of them into those transmission stages. So transmission investment would be 25% in this scenario. If 2 gametocytes, if 2 of those transmission stages is the optimal number of transmission stages, there's lots of ways to get 2. You can infect 6 red blood cells and turn a third of them into gametocytes. You can infect 4 red blood cells, and turn half of them into gametocytes. The point is, if you're optimizing the number of gametocytes, you can do that in a lot of different ways. It doesn't require low transmission investment. And what we're looking for is a solution here, some selective pressure, that makes you choose this or something like this, or further down on this, on the left side here, chooses a strategy with a low transmission investment rather than one of these strategies with a higher transmission investment. So that's what we're looking for. So neither of these hypotheses could do the job. So then we started thinking, well, what other challenges do malaria parasites face? We started looking inwards, and inwards, inside a host, parasites are often confronted by competition from other parasite genotypes. And it turns out you can show analytically that when there's competition, the parasites that do best in competition are the ones that exploit more resources. Exploiting more resources means foregoing investing in transmission stages and staying more often in this cycle of asexual replication. So Laura Pollitt was a PhD student with Sarah Reese, and she had some data that could sort of test this hypothesis that within-host competition generates an advantage to parasites that invest less in transmission. And so what this is showing you here is for days post-infection in rodent hosts in the lab, she infected them with malaria parasites of a particular genotype, shown in red here, and she's estimated transmission investment over the course of the infection. And the solid line shows you what parasites do when they're by themselves in infections, and this dashed line here tells you what parasites do when they're in competition with other genotypes. And you can see there's this wholesale reduction in transmission investment. And that was true for other parasite genotypes as well. In competition, parasites seem to adjust their allocation, their investment in transmission, and they reduce it. So they're doing it in the direction that our models predict would be adaptive in competition. Okay, so we've kind of already bridged some scales here. We've thought about the within-host environment, and then that R0 expression captured everything at the between-host environment. But like I said, the model is kind of treating hosts as bags of blood. And so Megan started thinking about this when she was a grad student. We're not exactly bags of blood. If we are bags of blood, we're dynamic bags of blood, and maybe there are processes at the within-host level that are important that this model has overlooked. So there's a bunch of changing resource availability. Red blood cells are being replenished by the host over the course of an infection. Maybe those dynamics are important. So Megan built a within-host model. I'm going to talk you through a little bit of the math. So she's tracking these uninfected red blood cells. They're being replenished at a particular rate. And then she's tracking these extracellular parasites that invade red blood cells, and then she's explicitly tracking these infected red blood cells as well. One convenient thing for modeling this system is that this cycle takes a particular amount of time. For the rodent malaria parasite that I showed you data from, that amount of time is 24 hours. So that makes it convenient to model this process with a particular delay in this case. So Megan's used a delay differential equation model. So there's a delay of 1 day for this process to finish. And then some proportion, in this case C, of all infected cells head towards making these gametocyte stages. And these gametocyte stages take their own amount of time to develop and be mature and be ready to be infectious to mosquito vectors. So she's also explicitly tracking the density of gametocytes in the system as well. So I'm only showing you one equation here, and I'll walk through it a little bit. So this is tracking gametocyte densities over time in the host. There's some number of red blood cells some amount of time ago. These blood cells had to be infected exactly alpha G time units ago because we're tracking now mature gametocytes. So there's blood cells some amount of time ago. There was those free-living parasites some amount of time ago. They bump into each other, get invaded at a rate P. Some proportion C of those will go on to produce gametocytes, and they have to survive that whole process. And then there's some decay of those mature gametocytes in the system. So that's one equation, but there are clearly 4 equations in her model. And from those equations, she can generate dynamics of infection that look like this. So this is the age of infection over days, the abundance of various stages. So in blue is the uninfected blood cells. She has infected blood cells. And then she has these gametocytes here in yellow. This is with a constant investment strategy. So every time step, exactly 5% of infected blood cells are going to get shuttled towards making those transmission stages. So this is a constant transmission investment strategy. Even with a constant strategy, you get fluctuating dynamics of abundance of those transmission stages. I think there's 2 things I wanted to point out here. If you look at the yellow curve, it looks identical to the pink curve except for it's lower down, and that's because 5% of infected cells are making those gametocytes, and it's shifted to the right because of that delay in the maturation time of those stages. Okay, so what we care about for thinking about parasite fitness is these transmission stages and generating new infections ultimately. So Megan took these gametocytes and asked, well, the way that my model had worked, my PhD time model, was assumed that more transmission investment, more gametocytes was always better. And there's actually some data to define that more is better idea. So here, within-host gametocyte densities are translated into the proportion of mosquitoes, if they were biting, that would become infected. So early on, making gametocytes, low numbers of gametocytes, isn't very effective. You're not getting a lot of transmission out of that. You get accelerating gains, and then things saturate. So at high densities of gametocytes, making more isn't actually very helpful for the parasite, for generating infections in the vector. So Megan took that trajectory of gametocyte densities and basically fed it through this function. So instead of having a trajectory of gametocyte densities, she's going to give me a trajectory of infectiousness to mosquitoes. And that's what this curve looks like. So that's with the 5% investment. And what you can see is those gametocytes that were made early on are basically wasted. There was too few of them. You don't get any infectiousness to vectors. If we choose a strategy at the opposite extreme, so really high investment, 40%, there's 2 important shifts. The first one is you delay the peak of infectiousness. So this is a bit counterintuitive. If you increase investment in transmission, you don't get a lot of transmission until later. You delay the peak of infectiousness. So more transmission investment means it takes you longer to be transmissible, basically. It takes you longer as a parasite to be infectious. And the reason for that is, in this early phase, your option, I entitled this talk The Parasite's Perspective because I always say you, and when I say you, I literally mean the parasite. So yeah, apologies. So you, the parasite, you have 2 options. You can either go down the developmental route that makes 1 transmissible stage, or you can multiply and produce something like 8 to 32 of those asexual, those blue parasites I told you about. So doing transmission investment early on means you're foregoing a whole bunch of opportunities for exponential growth, basically, in huge numbers. So if you reduce transmission investment early on, you get a huge biomass much faster. And so even if you're converting a small percentage of that biomass, you end up being more infectious early on. So low transmission investment means earlier infectiousness. The other difference you can see is that later on, there's an advantage. The higher transmission investment ultimately means higher payoff in infectiousness later in infections. So those are the two key differences between high and low investment, which are going to drive every other result that I show you. Okay, so Megan's first question was, if we just maximize trying to get out of the host, like total potential for transmission to mosquitoes, what's the best strategy? And answering that question, it means basically taking the area under these infectiousness curves. We want to maximize cumulative infectiousness over the course of an infection using different parasite strategies. What's the best parasite strategy? We made some assumption here about how long infections last. She took this estimate from some literature source, so this is like maximum possible length of an infection. If your infection is 280 days, what's the best parasite strategy? And the answer is, for the parameter estimates she used for the particular human malaria parasite that she used to parameterize this model, the answer is 20%, which is pretty high. And it doesn't matter that it's high. We're going to see what direction it goes when we layer on top other processes. But what Megan has also shown, and I'm not showing you, but you're going to have to take my word for this, she can also show if you put competition on here, you see the same result that I saw in my analytical models, where you get selection for reduced investment in transmission, somewhere around 1%. If you layer on early immune defenses, which can be overwhelmed by high parasite numbers, then making lots of high parasite numbers is good for parasites, so again, reducing investment in transmission. So both of those processes, within host competition and early immune defenses, which are nonspecific, both of those things select for reduced transmission investment from that 20 that I just told you. The other thing we've done, thinking at the within host level, is ask, what happens if we drug treat these infections? Can parasites mitigate some of the effects of drugs by altering these investment strategies? So we've done this in two ways. In this paper, we basically took this same approach, asking, given that infections are going to be treated at a particular day with a particular dose, what should parasites do? So this is like they're constantly exposed to this exact same kind of environment that on day 8, hosts get treated with drugs with a certain dose. What should parasites do? And then we did it another way that some of you might be more happy with, where we let parasites think that they're in an untreated infection and then ask if we drug treat and allow parasites to change their strategies, what should they do? Should they change their strategies? So one was a more sort of, if you will, like a canalized trait. We allow the evolution of the trait. We assume they're constantly exposed to the same environment. What's the best thing that they do? The other one is more like real plasticity. We change the environment and ask them if they can do better by changing their traits. But we basically find the same thing using the two different approaches. So for different drug doses, we ask what's the best transmission investment strategy. So if they get no drugs, they do some strategy. That's their like happy environment strategy. As we start titrating drugs into the system, the model predicts that parasites should reduce investment in transmission. And again, that's because if they reduce transmission investment, they can have higher parasite biomass, and some of those parasites will make it through drug treatment. Once you get sufficiently high levels of drug treatment, that will be like a curative drug dose, then the opposite is predicted. Parasites should actually increase their investment a little bit. And so the way we think about this is down here, parasites should adopt some sort of reproductive restraint to try to wait out bad times or overwhelm bad times. And then once drug doses get sufficiently high, it's more like a terminal investment strategy where this infection is going to get cleared, the parasites have to get out of here. So take all your resources and turn them into transmission stages to try to get out of that host. So that's what the model predicted. And then our empirical collaborators actually did the experiment where they had rodent malaria again. So they had these infections going for, I think it was 10 days. And then they did a single day of drug treatment and looked at how conversion rates, those transmission investment strategies, changed using different doses of drugs. And this is roughly what they found. So with no drugs, the parasites from day 11 onwards had a particular strategy. As you increased the drug dose and started killing more parasites, the parasites respond by reducing transmission investment to a point, and then the dose was high enough where most of the parasites are getting killed, that transmission investment goes back up. So the data, amazingly, kind of fit with what the model was predicting, again, for the effects of drugs within a host. Okay, so what we've shown in those two papers was that parasites actually can do better with no canonical resistance mechanisms, just by changing this life history trait, they can actually mitigate to some extent the effect of drugs. So in addition to these classical resistance mechanisms, maybe we need to, me and others like me, need to think about possibly the evolution of these life history traits in response to drug treatment. And one consequence of this is in that low to medium drug dose area where there was selection for parasites to reduce investment to transmission, that means all else equal, we'll be selecting for parasites that are more virulent in untreated infections. Okay, so that was all basically within host processes and their influence on the evolution of this trait, which is expressed within the host. But then we're going to think about processes that happen kind of outside of the host. And the first one is thinking about host recovery. So before we just had infections that last 280 days. That is clearly a very strong assumption. What happens if we relax that assumption? And so this is what we had before. We were maximizing the area under this curve, maximizing cumulative infectiousness over the course of an infection. And we're going to layer on top of that some sort of constant recovery rate. So a constant recovery rate kind of assumes that recovery of an infection is, in some sense, divorced from the within-host dynamics that are happening. And I'm going to relax that assumption later. But for now, let's just assume that there's a constant rate of drug treatment. And so that is one way by which you could get sort of this constant recovery rate of an infection. And assuming a constant recovery rate gives you this exponential distribution of infection lengths. And so now we want to maximize cumulative infectiousness, basically weighted by the probability that an infection lasts a certain amount of time. So obviously what this has done is put a premium on early infectiousness. If most infections, as you can see in this distribution, most infections don't last very long, then any advantage to being more infectious out here is not going to be worth a lot from the parasite's perspective. Being infectious earlier is going to be important from the parasite's perspective. So instead of this 20%, we actually see that the best strategy is one that has a lower transmission investment. Lower transmission investment means shifting the peak of this curve to the left, which will put it closer to the bulk of the infection lengths we see from that distribution. So this is what I showed you before, the single infection optimum where all infections lasted 280 days. This is the co-infection optimum where all infections last 280 days, but there's competition. And then this is showing you, I'm going to show you, optimal transmission investment if we're maximizing total infectiousness out of a host as we increase the recovery rate. So increasing the recovery rate makes most infections last less time. So as we increase the recovery rate, we see selection for reduced investment in transmission. And again, that is so we shift the peak of infectiousness closer to the left. Okay, so then we wanted to move even farther outside of the host and think about what if we consider what's happening in the vector population. So this is, I think, the coolest part of this model because I think it's the most novel bit. So we wanted to know whether parasites could respond to changes in their availability of the vector. And we have reason to think that they could respond to this from some experimental infections in birds. So this experiment was super cool, but they don't have the prettiest graphs. So I'm going to try to explain to you their experimental design. So they have these really long chronic infections, malaria infections in canaries. So these infections are lasting 300 days. And then over the course of that 300 days, they did these feeding sessions. So they would expose the canaries to mosquitoes. They would expose them to mosquitoes, and then wait 3 days, expose them to mosquitoes, wait 3 days, expose them to mosquitoes. They did that in 3 different blocks at 3 different times during the infection, and they took these blood samples. So they took a blood sample, then exposed them to mosquitoes, took a blood sample, exposed them to mosquitoes. And what we're going to look at is the parasite densities within those canaries over these windows where they're being exposed to mosquito bites. So this is showing you for 2 of these, I think this is session 2 and session 3, showing you the trajectories of parasite densities within the host. The open circles are parasites in birds that were not exposed to mosquito bites, and the closed circles are parasites in birds that were exposed to mosquito bites. And importantly, you see that exposure to mosquito bites leads to these increases in parasite density within the host. So the weird thing about this system is they can't separate out those transmission stages. The unfortunate thing about this system, they can't separate out transmission stages from those asexual parasites, so this is just a total parasite density. but the parasites are doing something. They're responding to the probing behavior of the mosquitoes or the immune response in response to that probing behavior, and they're changing what they're doing. They're increasing their biomass in response to the presence of the vector. So if they can do that, like they're A, pretty smart, but B, maybe they could also respond to the abundance of the vector in addition to the presence, maybe also the abundance. So that was our thought. If they could recognize changing abundance of vectors, are there different strategies that do better when there's lots of vectors or few vectors? That was the question we wanted to ask. And to answer this question, we paired with Lindsay Beck-Johnson, who's a postdoc at Colorado State, who has these amazing models of mosquito population dynamics that are driven by temperature. We're heading towards the model where we can drive it by temperature. Right now, we're just doing sort of a phenomenological, what if we increase mosquito densities, or what if we decrease mosquito densities? But ultimately, we want to be able to ask, in different areas that have different patterns of seasonality, if we feed those patterns of seasonality into this vector population dynamic model, does that lead to predictions for different patterns of transmission investment in these different areas of the world? That's where we're headed. We're not there yet. So how we get there and how we got to where we are so far is we've taken this within host model that I showed you a little bit about. So this is tracking basically infectiousness over time given different parasite investment strategies. We feed that into a between host model. So we put in a single infected host. That infection lasts some amount of time based on this recovery rate. And then we track recruitment of mosquitoes into the mosquito population. So again, this would normally be a temperature-dependent thing. Right now, we're just saying, right now we're capturing this by habitat size. So bigger habitat. If you increase habitat, you get increasing inputs into the mosquito population. If you decrease habitat, you get decreasing inputs to the mosquito population. And then we have some transmission rate from infected hosts to mosquitoes given by this infectiousness function. And we're basically tracking the number of new infectious mosquitoes recruited in this model. So again, over the course of an infection, which is maximally 280 days, we have an extra few days here because once the last, after 280 days, it might still take a bit of time before the last infected mosquito is actually infectious because the parasite takes some amount of time to develop in the mosquito. So over this amount of time, what is the total number of infected vectors we get in this model when we use different transmission strategies within the host? So our expectations, if we have a constant mosquito population size, nothing should change. The best strategy should still be 12% based on this recovery rate. And the reason for that is this was always the implicit assumption in the model, that there was constant supply of vectors at whatever abundance. If we change the abundance, make abundance higher but constant, the best strategy won't change, but the fitness payoff for that strategy might change. So more mosquitoes means more infected mosquitoes, but the best strategy given a constant mosquito population size is still 12%. It doesn't change the fitness landscape from what I've shown you before. If we have a declining mosquito population, what that means is infectiousness down here is even, the payoff for infectiousness at late infection ages is even further reduced. There's an even greater importance to these early infection ages to take advantage of the vectors that are highly abundant early on. So what that means, again, is it puts a premium on infectiousness earlier. So we'd expect from our 12% before, we expect that when you have a declining mosquito population, it should select for even further reductions in transmission investment so that you are infectious earlier, even more infectious earlier. And then we'd expect the reverse to be true in a growing mosquito population. So here, if you have lots of mosquitoes at these late infection ages, any infection that makes it out into this late age will have an advantage if it has higher transmission investment, because I showed you higher transmission investment means this part of the curve is higher. So we expect then that the best transmission investment strategy will be higher than that 12% when we weren't considering mosquitoes. Okay, so I'm just plotting this for one recovery rate, but the answers are the same, qualitatively the same, regardless of the recovery rate. This is showing you that, so this is constant mosquito populations. It is the same as having not considered mosquito populations at all. If we have an increasing mosquito population, we do better by increasing transmission investment so that parasites can take advantage of the increasing numbers later on. And again, with a declining mosquito population, there's selection for reduced transmission investment for the reasons that I explained. Okay, the last step, the last bit of biology across scales that we wanted to include was some sort of epidemiological feedback. So what I mean by that is, here is my caricature of what an epidemic looks like. Apologies to those of you who do epidemiology. So these are infected hosts over time, and the important thing is here, as the epidemic is growing, most infections are more than replacing themselves. The infected population is growing, so that infections are on average quite young in this part of the epidemic curve. And then on this part of the epidemic curve, where infections are not replacing themselves, infections are just getting older, basically, and not replacing themselves, the average age of infection is going to be quite old. So here, in this part of the curve, there again is going to be an advantage to getting out of your host faster, as fast as possible, to take advantage of the big pool of susceptible hosts in the population. Whereas over here, it might be harder to find a susceptible host, so being able to stay in an infection longer might be advantageous. So the only thing that we needed to add to this model was some susceptible host population. So it's no longer just the single infected host we're putting into this population. We're allowing there to be a whole pool of susceptibles that are themselves getting infected, that are themselves recovering from infection, and may themselves be contributing to this cycle of infection through the vectors. So now we're asking the question, instead of over a 280-day infection, we're just imagining the transmission season lasts the whole year, and we want to know what transmission investment strategy within a host maximizes the number of new infections generated over the course of that year. So for the parameter space that Megan has explored so far, it mostly is restricted to this growing phase of an epidemic. So our simulations don't get us over the peak, over the curve. So basically we're asking what happens when you include a growing epidemic, the influence of a growing epidemic on the evolution of this parasite trait. So again, I said, in a growing epidemic, most infections are young. There's going to be a premium placed on getting out of your host as fast as possible. The reproductive value of early ages of infection is going to be higher than late ages of infection. So once again, early infectiousness is going to be what's important here. So getting earlier infectiousness means reduced transmission investment. And that's what Megan finds. So these are these purple dots here asking what's the best strategy to maximize the number of new infected hosts in the population. The answer is a lower transmission investment strategy than any of these other things that I talked about. What I find interesting about this result is that these epidemiological feedbacks kind of swamp out every other effect that I've told you about. So all of these purple dots, even if I plotted them for these other recovery rates, would be right around the same transmission investment level. So that means these subtleties due to recovery rates or due to mosquito population abundance get swamped out by this growing epidemic. Okay, so when I put in recovery rate, I said we're assuming there's a constant recovery rate that's basically divorced from what's happening within the host, which probably seems unreasonable. So we wanted to allow transmission investment, these decisions that parasites are making about transmission investment, to influence the duration of the infection. The problem is we have no idea what direction it goes. So does more proliferation mean a longer infection, or does more proliferation mean a shorter infection? I actually don't know, and maybe there's people in this audience who know that answer. The data exists, sort of. Never mind, that was a tangent. I'm going to reserve the tangent. The point is we don't know what direction it goes, so we tried two extremes. So we said, what if there's a relationship between transmission investment and recovery, such that the more you invest in transmission, the slower the recovery rate. So more transmission investment means longer infections. You can imagine what that's going to do to selection on transmission investment. Probably there's going to be a benefit to investing more in transmission if it means your infection lasts longer. The alternative assumption is that more transmission investment means faster recovery. So in fact, you prolong the duration of infection if you focus on proliferation instead of transmission investment. So we actually think that this is the more likely relationship between transmission investment and recovery. I can explain that later if people are interested, despite the fact that often theory makes an assumption that's more like this. So we think this one's more realistic, but we don't actually know, and it doesn't actually matter. I'm going to show you both responses. So this was the purple strategy from before, which I said was pretty much constant regardless of what recovery rate you looked at or mosquito abundances. And this is what happens if you assume this relationship, so that the more you invest in transmission, the shorter the infection. There's actually really serious selection for reduced transmission investment. Regardless of which one of these fitness metrics you look at, you get this really clear reduction in the optimal transmission investment strategy, almost as strong as this co-infection effect, which I think is quite pronounced. And then in the opposite case, where transmission investment prolongs infection duration, I said there's going to be an advantage to investing in transmission. That's what we see. We see these extremely high optimal strategies where parasites are investing a ton of their resources into transmission, and that is maintaining longer infections and is paying off in these cases. Okay, so the key point from here is, I told you that these epidemiological feedbacks could overwhelm everything, so the subtleties of recovery rates and mosquito population dynamics didn't matter. That's not true. That's not true when we have these trade-offs happening, when we have transmission investment determining the duration of infection. If there is a relationship between these two axes, if there is a real relationship, that has a huge qualitative impact on the best strategy for parasites. So answering that question, what is this relationship, is going to be extremely important for understanding both the evolution of this trait in general and also whether these other things, recovery rates and mosquito population dynamic, not recovery rates because this would be a recovery rate, mosquito population dynamics and epidemiological feedbacks, if those are important, matters. If those are important, whether those are important depends on whether there's a relationship between these two things, recovery rate and transmission investment. I feel like I said all that really fast, so when that wasn't clear, you can ask me, and I will try my best to clear it up. Okay, so the important thing is I think that we have shown that stuff within the host is really important. Ecological processes within the host are important for generating selection and for shaping the evolution of this particular parasite trait, but stuff outside the host can also influence the evolution of this trait, which is expressed within the host. And it turns out that some of our best methods for controlling malaria and perhaps other diseases are things that happen outside of the host, things like bed nets. Controlling transmission is one of our most successful ways of controlling malaria infection, which means that these control methods are probably influencing the evolution of this parasite trait as well. What's really interesting is that I think we've shown any control measure that slows the spread of malaria, so gets us out of this increasing epidemic curve space, anything that slows the spread might actually relieve one of these selection pressures that's keeping transmission investment low. So these control methods might actually be relieving that constraint on transmission investment, and by relieving that constraint, you might end up getting more transmission investment, which actually equates to clinically less severe infections. If they invest more in transmission, they're making fewer of those host-damaging parasites. Okay, so I showed you a bunch of within-host stuff that generates strong selection for reduced investment, competition, innate immune responses, and drug treatment, subcurative doses of drug treatment. We saw all of these other things that also select for reduced transmission investment, host recovery, declining populations, mosquito populations, and growing epidemics, all increase the value of early infectiousness, which generates selection for reduced investment. And then we have these epidemiological feedbacks that can overwhelm all those other processes. But again, any relationship between this trait that we care about, transmission investment, and the duration of infection is going to totally change the landscape here, and it's going to determine whether high or low transmission investment is important, and we have literally no idea. So we have to figure that out. So I'm going to end here with some math to try to impress you, and again, to remind you who did most of the hard work. I did this hard work, the decorating of that cake. Happy to take any questions you might have. Thanks. Go for it.
So thanks very much. Really interesting talk. You pulled an impressive amount of stuff together on a really important question. So you're the only one, I think, in the room maybe with the capacity to synthesize all of that right now. But just focusing on one piece, this business about the epidemic curve. So that predicts, I guess, that you would see in regions where you have epidemic malaria outbreaks, sort of on the edges of the home range of the thing, you would see sort of fluctuating selection, whereas in regions where it's endemic, you would see these things work themselves out. I mean,
Yeah.
adaptation pushing you.
Probably.
So I can't. So do data agree with the predictions in that regard?
So I don't think there is data. So what you would want is transmission investment from infections from all over the place. Is that what you mean?
Right. You want transmission investment in different regions, right?
Yeah. So.
And some idea about the variability, I guess.
Yeah. So in the first case, I don't know if Hernan and co heard that question sufficiently. Nah. Okay. So the question was, we showed this effect of the growing epidemic. So you might predict in places where you have like really seasonal epidemics, you'd get this fluctuating selection on this trait that we care about, and in places where you have sort of more constant transmission, you'd expect something different. So is there any evidence of different patterns of transmission investment in places that have these fluctuating epidemics versus more constant transmission? And my first response to that is I don't think we have good transmission investment data. And so like I said, the way that people have been estimating transmission investment is like flawed. So postdoc Megan showed using simulated data that you basically never get the right answer with the way people have been doing it. So I don't think we have existing good estimates of transmission investment. We might be able to use some sort of proxy for virulence as like the inverse of transmission investment. And we have thought about that. And so we've been trying to look at a proxy for virulence being like case fatality rates for people who get infected in different parts of the world. That is also flawed because people in different parts of the world are going to be differentially susceptible. So we had this idea, which we're trying to do, which is look at travelers who return back to Toronto, who left from Toronto, went to different places in the world, came back to Toronto with malaria. And there's actually hundreds of cases like that every year just in my city. And so we wanted to use those as sort of like as close as we could get to controls who don't have like genetic resistance mechanisms from, living in a population that has historically been exposed to malaria and shouldn't have, ecological resistance mechanisms, so acquired immunity. If we can try to remove those things from naive travelers who returned to Toronto and had acquired malaria in different parts of the world, we want to know whether there's differences in virulence in those infections. So this is a really circuitous way to get to the answer to your question, which is, I don't know. We're going to do something that's going to approach answering your question, I think. We're going to try to do that, but it's still got some issues with it, obviously. We'll see. Yeah.
So question, it looks like no matter how you're altering the artificial schemes you use to do the experiment, the parasite can always figure out the best transmission rate. Did you expect because they sensing some very simple things, or they somehow differ in the time scale of your experiments?
Right, yeah, so our models are assuming that parasites, in general, our models are assuming that parasites are really smart and know both what the entirety of their infection looks like, how long it'll last on average, and what mosquito population dynamics are going to do. Yeah, so our models are assuming that they're really smart.
But when you do the experiment, you are essentially cook up a scenario that probably they have never seen. So
yeah, so the drug experiment that I told you about. So the question is like, what are the parasites responding to in these infections to change their trait? And the drug, so in both the drug experiment and the competition experiment, I don't know. I don't know what they're responding to. In the drug experiment, those are parasites that had never been exposed to that drug before. So they shouldn't know what that drug is or what that drug does. But it's clearly damaging them and their kin in the infection. So they might be sensing something to do with like parasite death in the environment. So yeah, so they could be sensing something in the environment that signals that they're not doing really well. The competition case, I don't know, because it's not like, the drug's going in and exploding infected blood cells, and so you're getting a lot of junk in the bloodstream that they could actually sense. In the competition case, it's not clear whether like they're sending messages to each other in some way, some sort of quorum sensing situation. We don't know, but they clearly are doing something. They are sensing something, and they can discriminate kin from non-kin.
okay, so so is not evolution happening in the time scale that you do the experiment? It's more like
Yeah, in the time scale of the experiment, it's plasticity for sure. Yeah. Yeah.
So I think, correct me if I'm wrong, but if you treat with drugs, then you'll get evolution of higher virulence, right?
More likely to get treated. Yeah, I was talking to someone about this recently. I don't know if it was about malaria or another system, but I agree with you. There's quite an interesting possibility of There's an interesting possibility of feedback there. So you're generating selection for more virulent infections, but those infections are more likely to get treated. Do you just end up in this like, is that like a Red Queen sort of thing, or not? And I don't know the answer to that. I think it's a really interesting question, though. Yeah. Cool. Thank you very much.